My training particularly is in microbiology, of which then I did a lot of years of research in genetics, particularly bacterial genetics, but also got expanded into genetics of other systems, since there's a lot of overlap as far as some of the basic principles. Also taught some graduate level genetics classes, and that would get you into some other systems as well. As a geneticist, it really began to ring then that Neo-Darwinism has not answered some of the basic questions that genetics poses. In fact, it's to the point that if you took a college-level evolution class, they will not tell you hardly anything of what I'm going to tell you this evening, simply because it doesn't work for them to be able to present evolution and to present this material. Okay, now I recognize probably for a number of you, genetics is Chinese. Yes. Normally I don't have people that can read this in the audience, so that works better. But what it comes down to is I will work to try to make it a little more translated. We're not going to go into real deep detail, but we'll try to keep it so hopefully you can follow along. All right, what we are looking at to begin with is what would be kind of in broad terms called the tree of life. And that is presenting the idea that all life is arrived from what is identified or labeled as universal common descent, or Darwin called it descent with modification. We have to put the universal in there, though, because that's one of the things that clearly identifies Darwinism, is that at some point in Earth history, humans and dogs share a common ancestry. And humans and dogs and reptiles share a common ancestry. And humans and dogs and reptiles and trees share a common ancestry. So we have to recognize that ultimately, at some point in time in Earth history, by Darwinian view, everything shares a common ancestry back to the original type organism at the very beginning of life. Okay, so this is what I'm going to be referring to when I'm talking about universal common descent, and also when I'm talking about Neo-Darwinism. We have to make sure we're clear on that. We're not talking about just change of gene frequency. That's not what they really mean when they're talking about evolution. This is what is meant when we're talking about evolution. Okay, this is what it really is. It's universal common descent. So that's what we're going to be dealing with tonight. All right, this is the creation model that I will represent. And as you notice, there is no universal common ancestry. For example, humans have always been humans. Dogs, or the canine group, has always been the canine. Horses have always been horses. Trees have always been trees. Sharks have always been sharks. See, there is no universal common ancestry. Okay. Do you climb Mount Everest in a single jump? Do you do that? No, you don't do that. Anybody recognizes that. Okay? In his book, Climbing Mount Improbable, Richard Dawkins, which, by the way, this is probably the only book he wrote worth reading. Okay? He actually says some things in there that I agree with, and I'll explain those to you here as we go along. Okay, he says Darwinism is not a theory of random chance, because obviously he recognizes the problem with that. It is a theory of random mutation plus non-random cumulative natural selection. Darwinism solved the problem of climbing Mount Everest, if you will, climbing Mount Improbable, by breaking the probability up into small, manageable parts, smearing out the luck needed, going around the back of Mount Improbable and crawling up the gentle slopes inch by inch, million year by million year inch. So this is how he views Darwinism climbing Mount Improbable. Okay? Inch by inch, nice, leisurely climb, walking the back trails. You don't even hardly know you're going up. Okay? That would be his vision. All right? Only God would essay the task, the mad task of leaping up the precipice in a single bound. And I agree. That is true. Any designer capable of constructing the dazzling array of living things would have to be intelligent and complicated beyond all imagining. Couldn't have said it better. Totally agree. Completely agree. Now, what I find very interesting is that what Richard Dawkins has done is he says that random mutation plus natural selection equals God. That's what he's done. Because you either do it in one bound, which is how God can do it, or you got to do it by his process, which is random mutation and natural selection. Now, mutation. I don't have time this evening to talk really much about natural selection. We're going to focus strictly on mutation. Recognize, probably if you've taken a biology class, if you haven't, you probably recognize it simply from just your exposure by watching television and anything else, that this is a double-stranded molecule of DNA. And it has then nucleotides, we call them bases, thymine, cytosine, guanine, adenine, or we abbreviate them TCGA. It is the sequence of those bases that then form the alphabet of the DNA. You change the sequence of those bases, you change then the arrangement of the letters, just as you would be changing the arrangement of letters on a page. So when you change the arrangement of letters, you have the potential of changing then the genetic information. You have the potential of changing what the DNA is saying. So a mutation then we're going to simply define tonight as any change to the DNA sequence. It doesn't make any difference how it occurs. It doesn't make any difference what caused it to occur. It doesn't make any difference how big or small it is. We're just simply going to keep it very simple. Mutation is any change to the sequence. If it originally was a T and it's changed to a C, that's now a mutation. All right, so effects of mutations. Well, one effect is that you can change from WT, meaning wild type, that's your original DNA sequence. You can have a mutation that changes where now the organism is what would be identified as more fit. And we'll explain some of that more as we go through. Okay? It may be less fit. It's now at a disadvantage. Or it may have no noticeable effect at all. That's called neutral. Okay? So you can have any of these changes occur with a particular mutation. Now, beneficial mutations obviously are going to catch our attention tonight, because these are the mutations that will increase the organism's ability to survive. So clearly that's what's of interest to the Darwinist. Okay? The organism is now, in their definition, more fit. All right? It maybe has greater speed, so it can maybe catch the prey better. It maybe has better eyesight, so it can see the predator coming better. It maybe has ability to survive better in drier conditions than some other plants, for example. So it gives it an adaptive, survivable advantage, and we'll identify those as examples of beneficial mutations. And they do occur. Not going to deny that those occur. In fact, we'll give several examples of these as we go through. You can see, because they are beneficial, this, like I say, is what catches the attention then of the Darwinian model. This is what they want to live off of, is look at all these wonderful benefits we're getting from mutations. Okay, so this is how then we build the tree of life, if you will. Universal common descent, mutations bit by bit, walking up that gentle slope, the back of Mount Improbable, one step at a time, one mutation at a time, if you will, so that eventually changes occur and you get plants. Other changes occur and you get reptiles. Other changes occur and you get mammals, and et cetera, et cetera. This is what I would call the simplistic view of the proportionality of mutations. If we have effect where on the left side of the zero is a negative effect, on the right side of the zero is a positive effect, then we would have, okay, 50% of mutations then are positive, so they're good, 50% are bad. See, and this is a good genes stay, bad genes go type thing. Natural selection that supposedly takes care of all the bad genes, and this is what you have, is all the good genes left, and isn't this wonderful? And we just move forward, and evolution just accomplishes great things. Okay, that's not true. None of this is true. This is a much more accurate representation of mutations within populations. We see that on the left, virtually all mutations are identified as either in what's called the no selection zone, which means that their benefit to the organism or their negativeness to the organism is so minuscule that you cannot really identify the effect it has. So essentially they're neutral. Or where we marked in red, then those are at least negative enough that the organism will have enough loss of survivability that it will start dying out. It maybe creates some type of defect in the organism so that over time the population will lose that mutation because the organisms with it will simply die off. I want you to notice one thing. That's the beneficial mutations, but it's not even to scale. If I put it to scale, you wouldn't be able to see it. And there's a lot of confusion in the literature of just how many of all the mutations occur, how many of them are actually identified as beneficial. And usually it's at best, 1 in 1,000, 1 in 10,000, 1 in 100,000, depending on the organism, depending upon the selection process, depending upon how they're really studying, trying to identify what the beneficialness of the mutation is. So we see, at best, it's very minuscule. Also notice, where does it fall? It falls in line of what's called the no selection zone. In other words, they're usually very hard to identify. Now that doesn't mean they can't be, and again we're going to see some examples, but we see very easily that many times within populations, the beneficial mutations get lost. They don't get selected, they actually get washed out because they can't become established enough in the population for them to be frequently within the individuals in the population. So just because it's beneficial, it may be very beneficial, but that doesn't mean that the population then suddenly just adapts with it. It may get washed out. Okay, this brings up what I consider is a crucial aspect of this whole topic, and that is the difference between phenotype and genotype. Phenotype refers to the physical characteristics of the organism. Is it tall? Is it short? Hair color? Eye color? Those are all physical characteristics. That's all phenotype. Genotype refers then to the genetics of the organism. What genes does it have? What's the gene activity going on? What's the regulatory aspects within the organism? That's the genotype. I say this as critical because what we're going to see is so many times what happens is that beneficial mutations are often viewed strictly from the phenotype, and the genotype is either not paid attention to or paid attention to very little. In Richard Dawkins' book, for example, Climbing Mad and Improbable, he does a lot of discussion of phenotype, virtually never even considers genotype. And that will lead you astray almost always, because what's going to happen is you're going to misunderstand what's going on in the organism. So we have to make sure we recognize it can be beneficial, but what's happening phenotype versus genotype. Okay, as the example, malaria resistance. Don't have much malaria around here, so this is something we think about very much, but clearly in areas where they have high incidence of malaria, any mutation that gives you an increased resistance of malaria certainly can be viewed as beneficial. Here's an example where you have on the red blood cell a protein called the Duffy antigen. And as it turns out, when you lose that Duffy antigen, so you're what we call Duffy negative, you actually have a significant increase in your resistance to malaria because it interferes with the malaria's protozoa of getting into the red blood cell. And as far as I've ever seen, there doesn't seem to be any negative consequence of losing that antigen. Doesn't mean there's not, it's just we haven't necessarily, that I've seen, been able to identify a negative consequence. So one could certainly argue this is clearly a beneficial mutation if you're in an area where malaria is high in incidence. But what occurs, how it occurs is that you inactivate the promoter, that is the starting region of the gene making the Duffy antigen. And that's what then gives you the ability of your red blood cells not being affected by the malaria protozoa. Beneficial, certainly, but what's it at? It's at the expense of the action of a pre-existing promoter. See, notice the difference between phenotype and genotype. Phenotype, Definite benefit, I'm resistant or more resistant. Genotype, I've lost something. Okay? Mainland mouse versus Santa Rosa Island mouse or beach mouse. Different fur color. What has happened is that there is a receptor gene that is involved in the levels of expression of pigments. In this case, imulin is more dominant in the mainland mouse, making it a darker fur color. But if you live in an area such as light sand, then the lighter fur color may actually confer an advantage to you by making you more camouflaged. So a mutation then in this gene actually reduces its activity, and so now you get more of a different pigment, making you a lighter color. But again, benefit if it gives you a camouflage adaptation, but what's it at? It's at the expense of the action of this gene making more of the imulin, which is the darker pigment. Okay? DDT resistance in insects, certainly of interest to people that deal with insects all the time. There's several ways that insects appear to become resistant to DDT, but they almost all seem to involve the protein P450. And specifically, they involve a mutation or some type of mutation that causes then loss of regulation of this gene, so you have overexpression. So you make excessive amounts of P450. Again, and some other genes have been tied in with this also, because remember, if you know your genetics, one gene or one regulatory system often will control several systems, not just one. If you're an insect and you've been sprayed with DDT, this would certainly be a benefit. But again, what is it? It's at the expense of the regulatory system that pre-exists in the insect to control the expression of P450. So we're having phenotype, benefit, genotype at the expense of. Right? Another, increased bone mass. This is a big one. If you go online and you look at beneficial mutations in humans, this is going to be a big one. Okay? There are humans that have a mutation where they supposedly have an increased bone mass that makes them more resistant to bone fractures. It involves a mutation in the gene called LRP5. And in fact, many sites will claim this is what's called a gain in function mutation. Wonderful for Darwinism, in their opinion. Wow, this illustrates the mechanisms, the genetic mechanisms that Darwinism is based upon. Okay, what's happening? Well, LRP5 is made from the LRP5 gene. This is a regulatory system or a regulatory protein that will work to induce a pathway called WNT. This pathway then activates other processes in the cell so you get increased bone mass. Now, it is regulated by the product of the SOST gene. This has a negative regulation effect on the LRP5. So you keep it level lower. What, as often the case is, it's like an accelerator. Less of the SOST product, more LRP5 made, and vice versa then. So what we see is we control bone mass by the production of the SOST gene. The mutation that leads to higher bone mass is a mutation that interferes with the binding site of the SOST product so that it can't bind adequately to the LRP5 gene, and thereby you lose regulation. When you lose regulation, you have overexpression of LRP5, therefore giving you increase in bone mass production. As I say, it's been claimed that this is really beneficial. I have read some recent accounts, though, that this isn't totally without a pathology. There does seem to be some consequence of it occurring. But regardless whether it's a consequence or not, beneficial or not, what's it at? It's at the expense of the binding site for the SOST product to regulate the gene. So it's at the expense of a pre-existing regulatory activity. So there's no way this would be identified as a, quote, gain in function because you're knocking out a pre-existing regulation. Right? HIV infection. Certainly if you're someone that's exposed to HIV, this would be very beneficial. What we see is HIV will bind to a protein called the CCR5 protein on the surface of one of your T cells in your immune system and infect that cell. Okay? There are individuals that are resistant to HIV because what has happened is they have a deletion mutation that inactivates the expression of the CCR5 protein. So their T cell doesn't have that protein. Without that protein, the HIV virus will not attach and infect. Beneficial? Yes, if you're exposed to AIDS, absolutely. If you're exposed to HIV, absolutely. But what's the genotype? You can tell me, what's the genotype? The genotype is at the expense of the expression of a pre-existing gene making a CCR5 protein. So you're starting to get the drift here a little bit, where we've been misled because they're always talking about phenotype and they forget to tell us about the genotype. Oh, this is wonderful, benefit, yes, but what's happening at the genetic level? Okay? A few more. Human lactose tolerance. When you were a small child, okay, you had an enzyme made called lactase from the lactase gene. Okay? As you got older and moved into adulthood, for some people, there is a gene called MCM6, and it makes a regulatory protein that blocks the lactase gene, and you no longer make that gene, and you can't consume very much milk product anymore. So we would identify you now as lactose intolerant. The irony is, this is wild type. This is what humans originally were. Those of us that can drink milk are the mutant. So we're just a bunch of X-Men. We're the mutants. Okay? What has happened is, in those of us with Northern European ancestry, in that population, because they were often herders of cattle and such, it became very beneficial for them to actually be able to consume the milk product from their cattle. A mutation occurred in the MCM6 gene and inactivated it. So the lactase enzyme is made your whole life. It doesn't shut down as you enter out of, or as you exit puberty. Beneficial? Yes, if you want to drink milk, or if milk is a primary food source for you because of areas where you can't maybe grow other crops and such, then absolutely it's beneficial. But what's the genotype? At the expense of a pre-existing system. In this case, the regulation by the MCM6 protein. If you go online and you talk and you code in beneficial mutations for Darwinism, you will get every single one of these I just gave you, and none of them will tell you the story I just told you. Because they'll only talk about phenotype. Why? Because phenotype works perfect for Darwinism, but genotype, see, does not. Genotype now tells an opposite story. Here's why it tells an opposite story. Well, okay, let's do antibiotic resistance first. This is very, very common. I spent many years doing antibiotic resistance research. This is very common as an example of beneficial mutations. Okay, you have a population of bacteria exposed to an antibiotic. Some of them will survive. And one of the reasons they'll survive is because they have a particular resistance. That resistance can be in many different forms, but they have a particular resistance. That would be known as antibiotic resistance. This is often touted as evolution in a petri dish. You know, living example of evolution in action. But see, once again, what is happening? A little chart I made up. Okay, different antibiotics result in different actions in the cell, so it takes different resistances. But notice the key in each of those: loss of, elimination, reduced, lost, eliminated, reduced, lost, eliminated. The organism becomes resistant to the antibiotic by mutation. It becomes resistant by eliminating the target system. That's one thing that bacteria can do that you cannot do. See, bacteria are wonderful Darwinian machines in that they can make these wonderful changes And then when the antibiotic is gone, they just grow it back. It's like chopping off, your foot's trapped in the bear trap, and here comes the bear. What are you going to do? Well, you might try to chop your foot off in order to get away. Okay? For a human, that's still not going to be very satisfying, because even if you got away, which you probably really still wouldn't, because I don't think one-legged person could outrun the bear. But anyway, your foot's now gone permanently. But bacteria, see, they grow the foot back. See, so that's why they can be so versatile, and that's also why the medical community is constantly facing the challenge of brand new antibiotic and they're already developing resistance. Because they laugh at our resistance because, like I say, they're wonderful mutating machines. They just do incredible things mutational-wise. But every one of these, reduced, lost, eliminated, reduced, lost, phenotype, benefit, resistant to bacteria or resistant to antibiotic. Genotype, I knocked something out. I knocked a pre-existing transport protein, a pre-existing enzyme, a pre-existing regulatory system out so I could survive the onslaught of the bacteria or of the antibiotic. Let's put this into perspective as an analogy. I call it the house analogy. Right? In your house, you have your dining room and you have your rec room. And they're separated by a single interior wall. It's not a support wall, it's just a regular interior wall. Okay? Your wife, being the wonderful, city socialite that she is, she wants a bigger dining room to entertain more guests. So what do? You knock out the interior wall so you can give her a bigger dining room. You lose your rec room, but she's happy, and everybody knows if she's happy, everybody's happy. Okay? Beneficial, absolutely. But what was at the expense of? It's the expense of that pre-existing wall separating into two different rooms. Now, are you going to tell a carpenter, here's how you build a house. You knock out this wall. What would the carpenter say to you? He'd probably kind of look at it and say, okay, you little nuts up here, kind of lost it a little bit. But that's exactly what we're being taught in our evolution classes. Here's how you go from reptile to bird, dinosaur to bird, to amphibian to reptile, to fish to reptile. Or excuse me, fish to amphibian. You knock out an interior wall. What? Where'd the wall come from? Oh, well, now that's a little bit more of a challenge. Where the wall came from. That gets a little tougher. But yet, when we recognize, if I'm going to build a house, I've got to build a lot of walls. Now, I've had students say, oh, well, you just take one wall and you replicate it 4 times. Okay, that sounds like a prison, not a house. Houses have floors and ceilings and windows and doors and plumbing, electricity, carpet, all kinds of other things in them, not just taking one wall and duplicating it. Plus, you got to explain, well, where did the first wall come from? The examples we've given so far don't build a wall, they knock it down. And yet these are the classic examples in the literature of supposedly driving universal common descent. But I think you would recognize if I'm going to go from a fish to an amphibian, and an amphibian to a reptile, and a reptile to a mammal, I've got to do a lot of wall building. I can't base it just on knocking walls out. That doesn't do me any good. Fish don't have hair. Where'd the hair come from? See, fish don't have the same kind of eyes you have. Fish don't have the same kind of lungs or heart you have. Where'd all that come from? See, so you got to do a lot of wall building if you're going to do that kind of transition. Okay, there are some fruit fly genes out there that are really frequently talked about as wonderful examples of building a wall. But the truth of the matter is they seem to be just rearrangements of pre-existing gene portions, and they really can't identify what their function is. So if this is the best example you have, then clearly I think you would recognize you're in a pretty desperate situation. Right? I don't know if any of you have heard of the famous bacteria experiment where we're taking E. coli and been growing it for now years and years and years and years. It's a wonderful experiment. I wish I would have thought of it. I wish I would have done it. Okay, it's a wonderful experiment. It's given all kinds of very interesting genetic answers. But clearly there's been a very definite evolutionary perspective to interpreting the results. What they did was, now it's well over 60,000 generations, growing the E. coli at a steady temperature of 37 degrees, day after day after day after day, year after year. And they would isolate from this population mutants. And the mutants that they would isolate, they'd find some of them would have, on a relative scale, a benefit. They grew a little faster. They divided a little faster. They were able to utilize nutrients a little better. So they were now benefited by some kind of mutation compared to their original parent. But what has happened is these mutants are results of disruptions, knockouts, losses of. They knock out particular genes. They knock out particular gene systems. They knock out particular regulatory systems. Every one of them so far has been you eliminated something. You didn't put something in, you took something out. Okay? So that led the researchers to coin a phrase called antagonistic pleiotropy. And what they mean by that is essentially a trade-off. You gain a benefit by losing. And we've already shown some examples, even in humans, where you gained a benefit by losing. They've even admitted in their papers that the main driving force of their 50-plus thousand generation experiment is antagonistic pleiotropy, benefit by losing. And I have sat and listened to several professors of evolution talk about this, and they almost never, talk about this experiment, and they almost never go into detail of what it means by antagonistic pleiotropy. But yet the researchers themselves are admitting this is the primary driving force of most of the mutants that they've identified, is antagonistic pleiotropy. They've gained by losing. You knock out that interior wall. But that doesn't explain where the wall came from. That doesn't explain how you built the house. It doesn't explain anything except taking a pre-existing and getting rid of it. Is this climbing Mount Improbable? Do you climb Mount Improbable by getting rid of? That's called climbing down. That's not called climbing up. Is this really an example of Dawkins walking that nice, gentle slope upward? No. What you're doing is you're going this way, going the wrong way. So this leads us then back to, again, you have to remember genotype versus phenotype. Now, the most famous of the mutants to come out of this study is what's called the citrate-positive mutant, or the citrate-utilizing mutant. Anybody heard of that mutant? If you've heard of that, a few of you have, yes. After 31,500 generations, they isolated some mutants of the E. coli that were able to utilize or grow on citrate aerobically. Okay? Technically, this is a new species because E. coli, by definition, cannot utilize citrate in aerobic conditions. They have not officially classified this mutant as a new species, but this would be an example, potentially, of a new species. What we find is, though, that anaerobically, in other words, in the absence of oxygen, E. coli will express a gene called CYP, and that's the transport protein that brings the citrate into the cell. And it will utilize citrate anaerobically. The key is that when the E. coli is growing in the presence of oxygen, so it's growing aerobically, the promoter for the CYP gene is not activated, and so you don't get expression of the gene. You get no transporter. If you have no transporter, you don't transport the citrate into the cell, you get no growth. So it's not a matter of somehow the mutant made a transport protein. The transport protein was already there. It just was regulated so that it wasn't being made in the presence of oxygen. All right, what has been found is that the citrate mutants really only involve two mutations that are essential for the organism to be CYP positive or to be able to utilize citrate. One is induction of the CYP gene aerobically, which makes sense. We already know it's there. It's just a matter of can you get it to be turned on? Can you get it to be expressed in the presence of oxygen? The other then, which is a bit of a surprise, is overexpression of another gene called DCT, K. And I'll explain its function in a second. Okay, what they found is that you can activate the SIT-T gene by simply moving it to the location in the chromosome, so that it now feeds off of another pre-existing promoter. You're not making a gene, you're not making a promoter. You're just simply moving the SIT-T, and that happens in E. coli chromosomes all the time. They move stuff around all the time. You're just simply moving it where it's now being able to run off of what's called the FIMB promoter or the RNK promoter, or also E. coli has an insertion sequence. That's a piece of DNA that moves around all the time. And in this case, it's called IS3. And IS3 has its own promoter also. And if you insert the IS3 in front of the SIT-T, then it just runs off the IS3 promoter. So the first step in getting this citrate mutant is you need to rearrange the chromosome so that the pre-existing SIT-T gene runs off of a different pre-existing promoter, one that's not regulated so that it doesn't function in the presence of oxygen. So now you have SIT-T being made even when there's oxygen around, even aerobically. The other mutation, which is a little bit more of a surprise, involves a transport protein that transports succinate and other C4 dicarboxylates. The reason this is important is because the SIT-T transporter is what we call an antiporter, a co-transporter. When it brings SIT-T in, it has to transport out something such as succinate. So if you're going to bring in a lot of citrate, you've got to export a lot of succinate, which means you've got to bring a lot of succinate in to export. So what we find is that a mutation in the regulatory system of DCTA allows overexpression of the gene because you've lost regulation. So it's overexpression. We saw that previously in some other mutations. So now what you have is you have excess of the DCTA being made to bring in excess succinate so you can transport in a lot of citrate. So it's interesting how these seem to work together. Very intriguing combination mutations the organism has done, but what do we have at the genotype level? Pre-existing gene being relocated in front of a different pre-existing promoter and loss of regulation of another pre-existing gene. Where in all this did we build the wall? We didn't build any wall. All right? Another study, they worked to apply the principles being used in the E. coli study to a sexual organism, to a sexual organism. Bacteria are asexual. They just simply divide. Okay? So they applied this to a sexual organism to see then if they could get similar type results. Now, you can't go as fast because being sexual, you got to have gestation periods and you're not going to have generation times measured in minutes. As you with E. coli, generation times are going to be measured in days, weeks, and depending on the organism, sometimes years. So in this case, they went to fruit flies, which is the common model used when you're dealing with sexual organisms because they're easy to work with and you can get fairly frequent generation time or fairly fast generation times for a sexual organism. Okay, what they found was that after 600 generations of what they identified as a very strong selection, they could not identify any unconditional advantageous alleles, that is, systems of genes, any unconditional advantageous system of genes that became fixed in the population. Demonstrating that comment I made earlier, remember how small that beneficial line was, and even then it often did not get fixed in the population, it gets washed out. They conclude the paper by saying the probability of fixation in wild populations should be even lower than the likelihood in these experiments because in wild populations the pressure, the selective pressure is going to be even less. So they kind of threw up their hands and says, I don't know if we can demonstrate, a Darwinian type approach in our fruit flies. We're just simply not seeing it. Trying to apply the same principle as was being used for the E. coli experiment. Now, one of the favorite things that you'll be taught in evolution is that an organism has a gene, we'll identify it as gene A, and every once in a while it'll just inadvertently duplicate the gene. So it's gene duplication. The reason they like this is because you see the organism now has a copy of gene A that will continue to serve that vital function that gene A serves, but the second copy you don't need. So the second copy we now are free to fiddle with, do all kinds of mutations to, and voila, we come up with gene B, and we've evolved. Okay, and that's the classic scenario you're going to be taught. But my response to that is, okay, give me your mutation examples. In other words, yes, you may take gene A and mutate it, but where's your examples based on what we see mutations do, the genotype, not the phenotype, where's your examples of building the wall? Give me examples of building the wall, not tearing the wall down. Beneficial, and evolutionists just go ballistic when I say, beneficial is really not that important. Like, oh, of course it is, it's the whole thing. No, because beneficial is based so much on phenotype. Tell me the genotype. Well, I typically don't know the genotype. Well, that's kind of important. What's going on at the genetic level? Are you building the wall? Are you climbing Mount Improbable? Or are you coming down? See, even if you came up with an example, said, here's an example where we're moving up Mount Improbable. Here's an example where we built the wall. What do we have? What then? You've got one example versus the hundreds and hundreds and hundreds of thousands of other mutations that occur all the time. What are we up to now? Known mutations in humans, 106,000, something like that. Known mutations in humans. The human population in the world is what? About almost 7 billion. Yeah, you have a little over 6 billion nucleotides in your chromosome. Every generation has every possible human mutation that can occur. Every generation. Are we becoming supermen? No. I think, in fact, we can make a very strong case. We're sliding down off of Mount Improbable. We're not going up. Okay? Even if you found that one classic example that I would have to say, yeah, okay, that does build the wall. That does climb Mount Improbable. You got one example. But in the meantime, you're going to what's called catastrophic error catastrophe. You're going through a genetic meltdown. So what is happening is that you're taking one step forward and 1,000 steps back. Now, how long will it take you to climb Mount Improbable at that rate? Yeah, you never would. And that becomes key. See, when you look at the genotype, you recognize the genotype is not working the way that Richard Dawkins, for example, was wanting to suggest. The genotype does not climb Mount Improbable. The genotype is coming down off. So this brings us to a little comment made in the book, What Darwin Got Wrong. Now, first off, recognize this book was not written by creationists. They said, okay, so if Darwin got it wrong, what do you guys think is the mechanism for evolution? Short answer, we don't know. In fact, as far as we can make out, nobody knows. See, when you look at the genotype, suddenly the picture isn't very pleasant. Suddenly, all these wonderful examples you have in the textbook, all the wonderful examples that are touted on the websites and such, all disappear. Because the genotype tells a totally different story than the phenotype. So now it's becoming popular to talk about evolution by loss. Evolution by knocking out that wall. Yeah, and that is true. You can get varieties, you can get variations, you can get changes in the organism if you knock out the wall. There's no doubt about that. But what do you have when you knock out the wall? You took a pre-existing and eliminated it. Does that climb Mount Improbable? No. Right? This is antagonistic pleiotropy. That's why, in fact, there is what's now called the third way. You can go online. It's thirdway.com. This is a group of evolutionists that are, a lot of them with genetic backgrounds, that understand everything I just told you. They understand it, and they say, yeah, Darwinism does not work. They're not going to become creationists because, see, to them, that's just too big a leap. They just can't deal with that. Most of them are atheists anyway. Okay? So they're saying there's got to be a third way. Now, they don't know what the third way is. If you read their website, they're saying, we don't know what the third way is, but we're trying to find a third way. Because they recognize that Darwinism doesn't work. They don't want to be a creationist, so there's got to be some third way. I guarantee you, if Darwinism worked, they wouldn't be doing this. But they know it doesn't work. So they're desperately searching for something else so they don't have to become a creationist, is what it really comes down to. So back to our universal common tree. Right? Which way are we going? See, what we see is we don't have the genetic mechanism to give us the middle part of the tree. We just have the mechanism to give us the variations at the tips, which we see all the time. We see those variations all the time. They're in the textbooks all the time, touted as evidence for this. But I guarantee you the evidence for this is not the same as the evidence for this. Everything I just showed you this evening fits within this. None of it explains this. Huge difference. Huge difference. We went back too far, sorry. Everything I just showed you also fits within this. Very nicely fits within this. What we have, see, evolution, climbing Mount Improbable. Creation says, no, we're actually coming down. We started at a high level at the point of creation, and we've been coming transcending down since then. Okay? What does our genetics tell us? Which one of these? See, genetics tells us we're not climbing, we're coming down. This brings us, just to finish up real quick, this brings us to what's called phenotypic plasticity. Now, this is a term that's used a lot in quiet. This is a term that's used a lot among professionals, but not discussed a lot outside of that sphere. Phenotypic plasticity recognizes that you can have very rapid changes occur to the phenotype, to the physical characteristics of an organism. Typically, the changes are limited. I would challenge, though, that even though paleontologists recognize its existence, they typically fail to account for it when they're looking in the fossil record. And I'll give you some examples. Okay? If we look at this, we say, okay, if we dug all of these up as part of the fossil record, which, by the way, much of the fossil record, like a vast majority of it, is shallow water marine invertebrates, shelled creatures. That's the vast majority of the fossil record. Okay. So if we dug up these shelled creatures as part of the fossil record, are these all the same species, or is this an evolutionary progression? How do we know? How do we know? Well, it depends upon our preconceived idea of what we want it to be. That's what it really comes down to. Okay? Now, we know for a fact that you can take many of these shallow water marine invertebrates, change temperature, change salinity, change nutrient availability, other environmental factors, and guess what? They morphologically change. Their phenotype changes. So when we have something like this that appears in the textbook, in fact, I took this straight out of a textbook talking about evolution. That's where it came from. How do we know this just isn't phenotypic plasticity? In fact, in 1985, the writer of this paper in the Biological Journal of Linnaean Society argued that fact, and he got poo-pooed. No, how dare you say that? Because we know it's evolution. We know it's Darwinian. He said, no, it could just be phenotypic plasticity. And the truth of the matter is, there's no way you can't say it's not phenotypic plasticity because you know this. So on what basis would you say it's not phenotypic plasticity? There is no basis. Now, if it's phenotypic plasticity, then you know it's not evolutionary lineage. And so there goes your example out of the textbook. Okay? We look at this, we'd say, okay, how would this be interpreted in the paleontological record? If all you knew about this organism was just the skull fossils you dug up, is this a different species? Are these 3 different species, 2 different species, same species? Is this an evolutionary lineage? We see this a lot in the literature where they say, oh, look at this wonderful evolutionary lineage. See, demonstrates how evolution works. Anybody have any idea what this really is? It's the bull terrier. It changed that much in less than 50 years because of selective breeding, granted, but it changed that much. If all we knew about it was just the fossils of its skulls, we would totally misinterpret what really happened. And I mean totally, emphasis on totally misinterpret. What we know is that in the DNA, certain events can occur, like you can have a repeated sequence and just put another repeat in, and that changes the phenotype. That changes the morphology of the snout, changes the morphology of the skull, changes the morphology of the organism. Or we can have repeated codons. For example, 18 to 20 repeats of the glutamate codon, or the glutamine codon, or 12 to 17 repeats of the alanine codon. Add in another codon in the repeat, and you change the morphology of the organism. This has been demonstrated over and over again. This is not building a wall. This is taking the pre-existing wall and painting it a different color, giving it wallpaper, giving it some other kind of texture. It's examples of how phenotypic plasticity can occur very quickly. Okay, so we see rapid genetic diversity can occur quickly within populations. Darwin's finches on the Galapagos Islands. Supposedly what these are, you see, is you have a number of finches with different size and shapes of beaks. And it's good reason to believe that at some point they came from an ancestral finch that was probably on the mainland, either Central or South America, that flew the 600 miles out to the Galapagos Islands and then began to diversify. According to what you'll see in the literature, they will argue this is an example of evolutionary progression over 2 to 3 million years. But what we've seen, for example, is in 1967, a population of finches were released on a different island in the Pacific. And in less than 20 years, they had spread to other islands, and they too had variations of shape and size of their beaks. And it was noted that it was comparable to the diversity of the finches we see on the Galapagos Islands. Now, did this take 2 to 3 million years? No, it took less than 20 years. So when you're interpreting the Galapagos Island finches, why would you suddenly impose 2 to 3 million years when you know it can occur faster? In fact, we now know with the Galapagos finches that what it is the differential expression. In other words, high level, low level, high level, low level of pre-existing genes. You're not building any walls. You're just simply taking what's already there and just altering how much is made. So you haven't climbed that improbable at all. And it fluctuates depending on if it's a dry year or a wet year. That's one of the pinnacles of evidence of Neo-Darwinism are the finches. And it has nothing to do with the claims of Neo-Darwinism. Right? Killfish will increase the number of the gill branches when they're infected with parasites. Not a significant phenotypic change, but certainly an example you have a phenotypic change occur because of an environmental condition, in this case, the presence of parasites. Right? Many of the smaller fish, minnows and guppies as example, in the presence of predators will actually remain smaller. They won't grow as big. I guess smaller, they can hide better. Okay, a 2-year study observed the physical changes in guppies and identified that the changes they observed in this 2-year period were the same changes that were identified by paleontologists as having supposed to take 2 million years. So it comes back then, wouldn't it be safe to say that paleontologists have very likely misinterpreted the fossils they're finding? Did it have to take 2 million years? Obviously not. We found example it can only take 2 years. Right? Red claw crayfish. Again, temperature and salinity can dramatically change the shape and size of the creature. If all we knew about them was some fossils in the fossil record, could we potentially misinterpret? Absolutely. See, this is what phenotypic plasticity does. It gives us a mechanism for very rapid but limited morphological change that, if not accounted for, would cause you to totally misinterpret what you're seeing in the fossil record. And I would challenge that's exactly what has happened. Okay? Stickleback. Oh, many times stickleback are one of the prime examples of evolutionary change, different speciations going on and such. And here we see, for example, sticklebacks were introduced into Lake Constance, and subsequently they were able to spread out from that first introduction into other streams. So they've set up their own ecology. Within 150 years, it's now been noted that they have, depending on the populations within the Lake Constance and the surrounding streams, different populations of stickleback from that original parental population 150 years ago that have changed in their armor, their size, and even their genotype. Classic example of what you see in the textbook under the chapters of evolution. If all you knew about these sticklebacks was in the fossil record, do you think they would likely be misinterpreted? Absolutely. Does this take 2, 3, 4 million years? No, 150 years, we already see it. Phenotypic plasticity. So what we have is a situation where we have demonstrated that the classic examples of mutations do not climb Mount Improbable. They don't build the wall. And we have demonstrated examples where simple genetic events can dramatically, or even maybe can dramatically or at least modestly change the phenotype of the organism, allowing the organism to be, or allowing then the potential misinterpretation of the changes being seen, so that it is not necessarily an evolutionary progression. In fact, the evidence we see would challenge that there's no such thing as an evolutionary progression as commonly interpreted. They can probably all, in some level, be ascribed to phenotypic plasticity. So I've given you reasonable and observable alternate interpretation to how genetics is viewed and how the fossil record is viewed. Millions of years is not needed for the observed changes that we see in these organisms. Therefore, we don't need to impose millions of years onto the fossil record. Okay? The mechanism does not require rare or unobserved mutations that Neo-Darwinism does. And it fits very well within a creation model, but it completely contradicts the standard Neo-Darwinian model. Hence the reason that one group of geneticists is looking for the third way, because they understand this. But I can't tolerate a creation model, so I got to go somewhere else. So there's got to be a third way. So that brings that portion to an end. All right, thank you. I guess we'll open it up for a few minutes for any questions. I don't know if you have a microphone that, or they just yell loud or something. Oh, this guy? Oh, all right. Okay.
Does anybody have a question? Well, I have a question. How do you, it's easy to empirically observe the phenotypical changes, but how do you measure the genotype changes?
Well, we're getting better, of course, at genotypic measurements because we're getting better at DNA processing, essentially. We're able to sequence much faster. We're able to do comparative genomics much faster and such than we could even just 10 years ago. So as time goes on, more and more organisms, their genotypes will be mapped. There's still a number of organisms who we don't have the genotypes mapped yet, so we don't know their genotype. And so it's a situation, okay, we had a change occur, we don't know what's happened. But that will become less and less. We will identify more and more what is going on. So
that's
I
more
tried to pick examples from the literature that are used where we do know what's happening in the genotype, because it's kind of futile for all of us to stand around and argue about what's happening phenotype-wise if we don't understand the genotype. And I don't get into those arguments because it doesn't, what's the point? Because before we understood the SID trait mutant, for example, there wasn't any reason to really talk about it as far as what was really happening on a Darwinian level, because you didn't know what was going on at the DNA level. And they totally missed, even the researchers themselves would have made statements that really weren't based upon what's happening at the DNA level. Because again, you can say it's beneficial. You can say it's very advantageous for your organism. No question. But what is it? It's not climbing man improbable. It's not building that wall. And in many of the examples I gave, in fact, what you're doing is going the opposite. You're actually knocking stuff out.
Yes. Okay, Sean, do you need to come up to...
You can lift that piece up and take it with you.
Okay. You can tell I'm a rocket scientist. I don't know how to work anything.
Thank you. Dr. Anderson, thanks so much for coming out tonight. Sean Bonzoff, 1040 Church. Just have this one question. Do we see any, I think you called it phenotypical plasticity?
Phenotypic plasticity,
Do we see any of that in human beings? I mean, it's easy, okay, so we add a bunch of salt and we leave them here and boom, we see the crayfish, it's grown. But do you see that, or does science see that happening in human beings?
It probably depends upon how exactly you're wanting to define or identify it. One could probably make the point that the increased bone mass density would be an example of phenotypic plasticity. You could probably make that case. One of the reasons it's more difficult in humans is because at 21st century, we pretty much control our environment. We're not out having to adapt like we used to. And even populations where they are still adapting, we don't have a starting point. We don't know where they've come from versus where they are now type thing. So it's hard to make a measurement, and you don't have the study where, okay, let's introduce predators into this tribe and see what happens to them in 10 years. That usually doesn't go over real well. So you're limited to things you can do as part of the experimental process. Probably if we use the physiology of similar animals, we could say that probably humans would have some similarity also, because we do see phenotypic plasticity occurring in similar animals. But it's interesting, we see it more frequently in those populations that I would say are much more vulnerable. The shallow marine creatures, invertebrates, the guppies, the minnows and such that have to do a lot of adapting or die. And humans don't face that near as much, so it's not as critical, so we don't see it as much.
So the lower on the food chain.
It does, to me, it seems to be that way from the examples I've seen. And again, those are typically the ones that are being studied. You have faster generation times. If you're studying elephants, you see it's kind of hard to get too many generations because they're very long generation times, long gestation periods and such. So when you're a scientist, you like to publish. And so you're not one to wait 20 years for your first paper. See, so you're usually studying organisms that give you fairly fast results. And that's going to be the faster growing, faster replicating, faster population exploding populations. So that's why you don't typically do as much of this kind of work with the slower, longer generation time organisms. But I would say we saw some examples I gave of humans. Perhaps in some sense you could identify all of them as phenotypic plasticity, but again, depending on how you want to define it. But clearly, at least to the point of we can identify mutations occurring in humans and see what's happening to the phenotype. And as we understand more of the genetics of human, we would then understand more of what's happening at the genotype as well. See, just remember, it wasn't but 20 years ago that we couldn't really even identify very much of what was happening even at the genotype level. We simply didn't know. And recognize again, and this is nothing against Charles Darwin. He gets picked on enough, maybe deservedly so. Darwin knew nothing about this stuff because nobody did. Now, one thing that's interesting, you've heard of Gregor Mendel. Please, hopefully you've had enough biology, you've heard of Gregor Mendel. Okay, Gregor Mendel did his pea plant experiments and worked out some of the basic principles of hereditary genetics. There was an unopened copy of Mendel's publication in Darwin's library. So Darwin had it, but he didn't ever even open it. Now, even if he had opened it, it's doubtful he would have understood it because almost nobody at that time understood it. But I've always wondered what would have happened if Mendel would have come along earlier and Darwin later. Because one of my challenges, which obviously I get scolded for, but no one's really, in my opinion, been able to really counter it, is if you came up with the idea of Darwinism today, you'd get laughed at. Because we know enough genetics to know that's just nonsense. But because it's so established, it's so entrenched, and don't underestimate the value of entrenchment into the scientific community. It's so entrenched that everything has to work through it, whether you got to pound it in with a hammer or not. You got to work through it. As opposed to introducing as a fresh idea, they say, well, that's silly. Doesn't begin to fit. It's a total different situations going on. I think he's got a question. Then if you want to come forward to the mic, maybe?
Only because we're streaming and we want people to benefit from hearing the question.
Speak it out, he could repeat your
Yeah, I could repeat it. Yeah, that's true. Yeah, if you just speak it out, I'll repeat it. Yeah. Mm-hmm.
But nowadays we see that their roads are growing properly, and so there could be different environmental changes that could actually promote adaptation in different time spans. So how do you account for those?
Okay, I don't know if I can repeat all that. If I'm paraphrasing the question correctly, you're asking for an accounting of environmental changes that are occurring to the populations. Is that correct?
Right, so with the finches, we saw around the 1900s when Darwin first was observing that it's taken millions of years for evolution, and then in the 1962 or 70s, that's when they set finches loose again. But since the two different time periods could have had two different environmental situations, that could affect the amount of time that the finches had to adapt to their surroundings.
I would say that's absolutely true. And nothing in fact, that actually supports what I'm saying. That within the time period between when Darwin was there in the 1830s and when they started studying the finches in the 1960s, you have an unaccounted for period where changes could have occurred, but that's the whole point. You're talking about 130 years of changes, not 2 or 3 million years of changes. And absolutely, environmental, that's one of the things we talked about. Dry year, wet year, dry year, wet year, environmental changes, temperature, salinity, all of those are environmental factors. And they very much play a role in phenotypic plasticity. And you're talking about African Americans. I think that absolutely fits in also with this. You're not talking again about a 2 or 3 or 4 million year period. You're talking about a very short period of a century or two where you saw changes occur in the human because of certain conditions they were being exposed to, certain conditions they were having to deal with. That's phenotypic plasticity. So I think what you're saying actually is very in line with what we just talked about. See, none of that, again, took millions of years. Those are types of changes that are occurring very quickly. But if you didn't observe them occurring, and all you knew was just one point, like 2016, see, what's the interpretation? Oh, this took hundreds of thousands or millions of years to occur. And yet, if you were taking several points along the way, you'd notice, well, this has been occurring just in a very short period of time. And you got these changes in 2 years, or 10 years, or 20 years, or 150 years. But none of those changes are 2 million, 3 million, 10 million years. See, so absolutely environment has an impact. That's part of phenotypic plasticity, is the organism adapting to a particular environment it finds itself in. Remember, if we go back to, if we go back to our shelled creatures, what we find is that we can get this kind of change very quickly. It's not even a 10 or 20 or 30-year process. It's a very quick process. And yet, this is what's shown in textbooks as examples of millions of years of change. And notice, see, this is environmental. It's temperature, it's salinity. Those are very much environmental factors, because that's what phenotypic plasticity is going to deal with, is the organism responding to. Now again, see, the environment is not giving the organism something new. You're responding to that. So it's already a programming within the organism. It's not the environment causing a new program. So phenotypic plasticity is the organism responding to the environment it finds itself in. So to say everything you said fits very well in this. We can talk later if you're still a little confused about that, but everything you said, as far as I see, is exactly what we're talking about here. Any other questions? Yeah.
All right, so I'm not going to kid you, a lot of this goes right over my head. But I'm curious because a couple times you said they're almost willing to believe anything but creation. They're looking for something so that they don't have to go with creation.
Mm-hmm.
So my question is, how much pressure is there in your experience for true scientists and people that truly love scientists have got to know on some level that these books, that the textbooks and stuff are, I'm just curious, how many of these teachers and professors and scientists are, do you think are hip to that?
That kind of opens up a whole broad topic, obviously. I'll try to keep it very narrow, because that in itself is two or three presentations on its own. Obviously, because you're talking about sociology and human behavior and everything else. The first thing you recognize for any of us is humans, we often don't understand things. Whether you're a scientist or not, we don't understand. And yet, sometimes, does our not understanding stop us from jumping ahead anyway? Plowing straight forward? The publication, The Conversation, a couple years ago, had a very interesting article in it talking about that many ideas in science are driven by what's called the herding effect, which means that even when there is and this is what they said in the publication, even when there is a absolute motivation to be totally objective and seek nothing but the truth, you still, more times than not, will base your conclusions upon your peers' behavior than upon the data. And what this article concluded was that the herding effect often has the effect of driving scientists to an incorrect answer. Now, that doesn't always occur, obviously. I am a scientist. I believe very much in the scientific method as a means of understanding the creation, understanding the world around us. But within that context, I recognize that if I am presenting at a scientific conference about how a certain enzyme folds, chances are there's only 7 people in the world that care about it. And we may get in a real passionate argument, but you know what? The New York Times isn't there recording it. They could care less. But if you have a conference where they are going foot and nail about something that is much more worldview, where'd you come from? What's your purpose? What's the meaning of life? Those types of things. Suddenly, then the press is right there too, and I would also argue that objectivity starts dissipating away, and you become more and more opinionated on and the herding effect really comes into play. See, when you're a graduate student, it's assumed you are going to not only have the same evolutionary values as your professor, but you're going to have the same political and moral values also. And if you don't have the same political or moral values, it may not mean you're going to get dismissed, but it may mean you're going to feel kind of isolated and even sometimes openly ridiculed. You believe that? I didn't know anybody alive believed that anymore. That's the herding effect. Herding them in, herding them in, herding them in.
Quick follow-up on the same question. How can you even have a reasonable conversation who says the Earth is a million years old? I mean, how can somebody prove, much less disprove that? You can't even have a rational conversation.
We're going to talk a little bit about that in my next presentation. I'm not going to go real heavy into age of the Earth because that's not my specialty. But we're going to touch on things that would be used to help identify what the age of the Earth is, and different people would have different opinions about what's valid and what's not valid as far as evidences for the age of the Earth. Clearly, the evolutionist would have what they would consider tests that demonstrate great ages. You know, this is 70 million years old, this is 300 million years old, and they have tests that they would say demonstrate that. I would challenge that I don't think your test is that accurate, and so then we go back and forth on what really is or isn't valid. But see, there again, that herding effect can come into play because, see, if you don't accept my interpretation of the age of the Earth, all of a sudden now, well, obviously something's wrong with you. And so then it becomes difficult to even get them to listen to you because then it's just a matter of, well, clearly you don't understand and you're not worth talking to.
Well, it's arguments of faith, and you're not going to prove it.
Well, there is some of that, clearly, because you're talking about things that have happened in the past, and so we can't reproduce it. All we can do is attempt to reconstruct it. Yeah. So, but don't ever underestimate peer pressure. Unfortunately, because we are humans, we're subject to it. And clearly in this day and age, Darwinian evolution is so entrenched. And I see Joseph brought up the movie Expelled. Now, obviously, again, the movie Expelled received a lot of criticism for supposedly being biased, which I always thought kind of interesting because the people criticizing it are some of the most biased. In fact, I've been in the university settings off and on for most of my professional career. And there's one thing I would go on record as saying, and that is that the universities will pride themselves in their openness, their tolerance, their acceptance of diversity, and there's few places that are more intolerant than a university setting. You just try to maneuver out of their little narrow concept and see how tolerant they are. One thing I found about the tolerant movement is it's tolerant of views that it agrees with, and highly intolerant of views it doesn't agree with. I'm sorry, but when I look at my dictionary, that's not the definition of tolerance. I find that very interesting and rather hypocritical. We're tolerant of everything as long as we agree with it. Well, that's not tolerance. That's not tolerance at all. And you'll find very few places in science that are less tolerant of ideological diversity than university settings. And it's not by chance. This is where the future scientists are trained. At Arizona State, there are many great scientists, many great future scientists that are being trained right here, right now. But we have to recognize this is a very ideologically intolerant setting. And they better toe the line. Hurting, hurting, hurting. No?
Let's take a short break, and then we can start our second presentation, Brad. It's like 8:20 right now.
Yeah, something, yeah.